Okay, so it's 9 o'clock. I think we can get started. Welcome everyone to join us for today's Immune seminar. So it's my pleasure to welcome our today's speaker, Dr. Xiang Zhao from Center of Excellence in Molecular Cell Science, Chinese Academy of Science. A very brief introduction for Xiang. So Xiang actually currently is a fresh PI in Chinese Academy of Science in Shanghai. So Xiang completed his undergraduate study in Fudan University in Shanghai, and then further pursue his a PhD in National University of Singapore in immunology. So from 2018, he actually after he completed his PhD training, he moved to Stanford to get trained with Christophe Garcia on TCR engineering for to complete a series of really great synthetic biology and T cell biology work. So from just last year, after he completed his postdoc training, he moved back to China to start his independent research program, as I just mentioned, in Center of Excellence in Molecular Cell Science. So Xiang's lab mainly interested in study T cell receptor engineering. So as we probably are aware of that the T cell receptor binding ligand peptide MHC is the molecular basis for the immune system to recognize different antigens. So Xiang's group want to try to comprehensively use immunology, protein engineering, and the other method, particular T cell display library technology and high throughput functional screening technology to understand the molecular machinery of T cell receptor signal transduction and T cell receptor signaling, and try to use these idea and knowledge in the cell therapy. There's a couple of different research focus in Xiang's lab, which including try to target tumor antigens and try to transform T cell receptor with high signal intensity, physiological affinity, and no off-target toxicity, and then try to study their immunological mechanisms and applications in different disease models. Now, the other focus part of Xiang's lab is try to engineer immune receptor ligands with new phenotypes and try to identify new immune receptor ligands. So as I just mentioned that Xiang actually completed a very fruitful both PhD training and postdoc training with high profile publications. With these, he actually received numerous awards. I think the most recently one is he got Stanford Dean's Fellowship and also Stanford BioX Interdisciplinary Initiative Seed Grant Program. So I think he's still going to talk about what he's most interested in, and then probably some of his postdoc work, which is the TCR signaling regulate by catch bounds. So thank you, Xiang, and then please go ahead.
Yeah, thanks, Chuan, for the nice introduction. Good morning, everyone in China, and good evening, everyone in the US, and for the whole world. Thanks for attending today's seminar. I'm Xiang Zhao from Chinese Academy of Sciences. So today I'm going to talk about the mechanism of effective T cell signaling regulated by catch bounds. So I'm a junior PI, so I just opened my lab last year in June at Shanghai Institute of Biochemistry and Cell Biology, Chinese Academy of Science. So the lab is located in Shanghai, China. So it's a really amazing institute with lots of resources. So highly recommend for any talents interested in doing research in Shanghai, China. So I mainly focus on this molecular basis interaction between T cell receptor and peptide MHC. So it's really an amazing system about recognizing the foreign antigens. So we know that we actually meet with this challenge from environment full of pathogens and these dangerous signals, for example, virus, bacterial infections, and sometimes also these tumor cells rising in our own body. So our immune system need to have a way to recognize all these foreign antigens. So one of the very elegant system is that those proteins from like virus, bacteria, or tumor cells, they can be engulfed and digested in these antigen presenting cells. So they can have a way to process those foreign proteins into a pretty short peptides, like within 20 amino acids. Then the cell have this machinery to load this short peptide derived from these foreign pathogens or tumor cells to load on this molecule called MHC. So MHC is kind of a membrane protein, so they can present this peptide information on the surface of antigen presenting cells. So this is one of the very important systems in our body to really present those danger signal from those pathogens or tumor cells on the surface of these antigen presenting cells. So the next step is really like how our body really sense the presence of these peptides. So the peptide MHC molecules have their own very special receptor called the T cell receptor. So as its name suggests, the T cell receptor is expressed on the surface of T cells. So T cells really one of the central role in our adaptive immunity. So T cell use the T cell receptor to recognize those peptide MHC. So the T cell can sense this danger signal from the pathogens or tumor cells. And after this T cell receptor binding to the peptide MHC, the T cell can transduce downstream signaling to activate the T cells. So in my last 10 years, I'm mainly focused on this interaction between T cell receptor and MHC. So the downstream signaling for the TCR is really complicated. But it's also very important basis for this activation and differentiation of the T cells. So in the last 10 years, I'm not only focused on this interaction between PMC TCR and also look at this downstream signaling, also how this structure between T cell receptor and MHC could tell us. So in my last 10 years journey, I really need to thanks to my supervisors. For example, my PhD supervisor Nicolas Gascoigne. So when he did his postdoc with Mark Davis, they cloned this mouse TCR genes. So I think it's one of the pioneer work in this field of TCR interaction with PMC. I also did my postdoc training with Chris Garcia, and Dr. Chris Garcia actually did his postdoc with Ian Wilson about solving these mouse TCR PMC structures. So I think these two pioneer work is really the fundamental biology knowledge we learned about this TCR interaction with the PMC. So today I mainly talk about some of my previous work I did in Chris' lab about this, how this TCR, effective TCR signaling is initiated by this catch bond. So when I just got to Chris' lab, they just published a pretty interesting story about these effective TCR PMC interactions. So they found two very interesting examples. So there are two different TCRs called TCR5I9 and TCR55. They can recognize the same this danger signal from HIV peptide. Although these both TCR can recognize the same PMC, they found the strange thing is the TCR59 could be activated by the HIV peptide, but not TCR55. You can see in this signaling data, you can show that the TCR55 can be activated by the HIV peptide. However, after they solved this crystal structure between both TCR59 HIV peptide and also TCR55 and HIV peptide, they didn't find any very significant difference between these two structure complex. And they also measured the affinity between the same HIV PMC with different TCR, they found they have very similar affinity. So, and they also, both TCR can be well stained by these PMC tetramers. So they have investigated so many potential mechanisms, but it seems just like everything is similar between these two TCR and PMC systems. So they couldn't find a mechanism to explain why this TCR45 could bind to PMC with pretty high affinity, and it's a very obvious, clear structure, but the TCR45 couldn't be effectively activated. So later they look at the other alternative mechanisms. So almost the same, at the same time, they're inspired by another group about maybe the mechanobiology of the TCR can explain these differences. So it's not surprising that T cell actually is very athletic. When they're scanning the tumor antigens, for example, they are in very fast migrations. So actually T cell and this force actually originated from some cytoskeleton rearrangement. So it's not surprising that TCR need to sense the force, and actually TCR need to deal with this force presence to affect their signalings. And one of really interesting experiments to show that TCR can sense force is that if you coat some cover glass with these PMC molecules and with a hairpin, DR hairpin, you can see that after you adding a TCR T cells, you can see that the TCR actually can open this hairpin, DR hairpin with around like a 12 Newton threshold to stop this quench the fluorochrome. So it's a really direct evidence to show that TCR can sense the force from the internal cytoskeleton. And yeah, also some previous research suggests that this force could be originally from the cytoskeleton rearrangement to apply on the TCRs. So almost a similar time, another group from Dr. Chen Zhu at the Georgia Institute of Technology, they use a very special instrument called biomembrane force probe. So for the first time, they could apply a force on the TCR to mimic this fast T cell migration in physiological settings. And they measure this bond lifetime between TCR T cells and a PMC molecule coated on the beads. So they found pretty striking difference. For some, so they studied this in these OVA ultrapeptide ligands and OT1 TCR systems. So found that for some strong agonist, like OVA peptide, they form this kind of curve between different amount of force and the bond lifetime. So you can see when you, at first you increase the force, you actually elongate the bond lifetime between the T cell and the PMC. It's kind of counterintuitive because it means that actually the shear force actually help the TCR to engage with the PMC. But if you continue to increase the force, it will outcompete this mechanism. And you can see the force will start to disrupt this interaction between TCR and PMC. So they call this kind of curve as a catch bound, means that at first the force actually help the TCR to catch and engage with the PMC. However, for some weak or even antagonist peptide, you will see that the force only disrupt the interaction between TCR T cells and PMC. So you will see that when you increase the force, you will only shorten this bond lifetime between TCR T cells and PMC. This is also called a slip bond. So they think that this catch bond versus slip bond can describe two different kind of TCR PMC interactions. So back to this TCR55 stories. So Chris lab these investigate these catch bond properties for both TCR. So as a reminder, we just talk about these two TCRs and saying TCR55 could bind to the HIV PMC very well, but couldn't be activated. So they measure this catch bond phenotype between this HIV PMC and the two different TCRs, and they found that this stimulatory TCR59 could form this catch bond with PMC, but the TCR55 could only form this slip bond with this HIV PAPMC. So it's clearly, so this catch bond versus slip bond mechanism clearly explain why some TCR could bind to the PMC with pretty high level affinity, but could be inactivated. It suggests that the formation of catch bond is required for effective TCR signaling. And we actually also look at similar mechanism in another PMC class 2 system, and also found something new and very interesting. So it's mainly done by my students Lan Xinhu and also Nan Wang at Chris lab. So it's not published yet, but it's something very different from this class 1 system. So we also identify two different TCR, TCR6 and TCR11. They can engage with the same HIV PAPMC class 2. This DR4 molecule presents HIV gap peptide. So we solved the structure, the two structure between this DR4 HIV gap with two different TCRs. So that this TCR6 can engage with PMC class 2 in a very canonical docking mode with pretty high affinity, and the TCR6 could be activated, and the TCR6 can form a catch bond with this PMC class 2. However, for this TCR11, we actually at first found that they could bind to the PMC class 2 with pretty high affinity, but couldn't be activated. And not surprisingly, the TCR11 formed this slip bond, not catch bond with this PMC class 2 molecule. So we at first think that the reason for this inactivation is because of this slip bond formation. But after we solved the structure between TCR11 and the DR4 HIV gap, we found actually TCR11 they interact with PMC or dock on the PMC class 2 in a reverse docking mode. It means that compared to this canonical docking mode, the TCR11 reversed around 180 degree to engage with PMC. So it's actually firstly described by Jamie Rosengren and Nico Lagrota in Australia about this reverse docked TCR PMC interactions. But we think that the importance of this reverse docking is maybe because in this way, you couldn't form the catch bond at first. So you always see this ineffective TCR signaling between reverse docked TCR and PMC. So we think that maybe the mechanism for explaining why you couldn't find you rarely see this interaction, also no signaling between this reverse docked TCR and PMC is because they always have this no catch bond formation at the beginning. So we're still wrapping up this story. So let's back to this TCR55 story. So we kind of summarize these results and found that the only important difference here is that whether the TCR could form the catch bond or not with the PMC. So you have effective signaling or not. And it's a really good example because the two TCR actually have similar affinity. So it suggests that maybe the TCR PMC interaction could be engineered without altering the affinity, but just significantly enrich the catch bond. You could still enhance the TCR signaling or induce the effective TCR signaling. So that's where we have this idea about catch bond engineering. We think that compared to is a traditional method called high affinity maturation to engineer TCR with better TCR signaling. They really are under this increasement on the affinity. We think that we can enhance this TCR signaling by enriched catch bond, and you could probably also keep the affinity at a similar low level. So that's what we call a high potency, low affinity engineering for the TCR. And we think that it's would be really useful for tumor TCR engineering. As we know, most of tumor TCR, they recognize those self-peptide as antigen. but with pretty low binding lifetime and very low potency. We think that maybe this cache bond engineering idea could enrich some cache bonds between this tumor-reactive TCR and PMC. So in this way, they can engage with longer TCR-T cell bond lifetime. So you will have this enhanced effective TCR signaling. So, so we use this TCR55 as example for this engineering. So as we just mentioned, that the TCR55 can bind to this HIV PMC with pretty high affinity. We solved the structure, but the TCR couldn't be activated. So we want to engineer a TCR55 mutants, which the same PMC could activate the TCR55 mutants. So based on this structure, we firstly need to design a TCR55 library. So the idea is like after this library engineering, we could introduce some new cache bonds between the PMC and the TCR55 mutants. So we think this the model basis is that we need to enrich some new hydrogen bonds or salt bridges, which are frequently discovered between the cache bond formation. So by looking at these structures, we actually select some residues are around 4 angstrom distance from this PMC, because we think the distance of 4 angstrom won't be too far away for those TCR residue to have this new interaction engage with PMC, and also not too close to affect these specific PMC interactions. So we select around 3 or 4 residues on both either TCR alpha chain or TCR beta chain. And then when we establish these libraries, we didn't try to randomize each residue into all 20 amino acids. We actually only limited them into charged amino acids or polar amino acids, because we think that those amino acid species have the best chance to form new hydrogen bonds or salt bridges. So in order to engineer and select this TCR 55 library, we also actually established this new method called T-cell display TCR library and functional selection. So it's pretty different from what people did in the field previously. They usually display this TCR or antibody library on the surface of like ribosome, E. coli, or phage or yeast, and can only involve these proteins by this high affinity maturation. We actually, for the first time, display this pretty big TCR library on the surface of T cells. The reason we want to display the library on the surface of T cell is that later we can use the T cell downstream signaling as a parameter to selection for this library. So that's what we did. We cloned this TCR library, and the library size could be like 10 to the power of 8 different TCR in the library. After we cloning and made this lentiviral library of the TCR and infect the T cells, now we have this T cell display TCR libraries. And we use the antigen binding cells to present, for example, HIV peptide to activate the whole the T cell display TCR library. And we did 2 things. We stain the library with PMC tetramer and also the activation marker C69. So because we want to see if this if we actually can evolve TCR by enriching cache bonds, not high affinity maturation, to enhance this effective TCR signaling, we actually redefine this evolution pressure by high level CD69 staining, but a low level PMC tetramer staining. It means that we only want to functionally select high level signaling, but low level or similar low level affinity, binding affinity to the same PMC. So it actually was a pretty effective strategy. So by doing this on this TCR55 library, we were able to enrich a population of cells with high level CD69 staining and also still low level this PMC tetramer staining on both alpha chain library and beta chain library. So we look at those clones from the selection. So we actually identify multiple TCR55 mutants which could be activated by the same HIV antigens. For example, here we show that the Y-type TCR55 couldn't be activated by the HIV peptide, but we identify mutants like this TCR55 alpha chain at residue 98. The Y-type residue was alanine, the mutant to histidine. We found that this histidine mutant actually could be highly activated by the same HIV antigens, and also some other mutants with different amino acids mutation at the same residue. We actually measure the affinity for all these mutants between these mutants and the HIV antigens. And we found that there's no correlation between the TCR activation and the binding affinity, suggesting that we were actually not really doing high affinity maturation for those mutants to enhance their effective signaling. So the mechanism is that is the catch-bound enrichment, because we select several residue from these or several mutants from this group, and measure these catch-bound properties between different monofolds and their bound lifetime. So we found that they all form pretty significant catch-bound curve between different monofolds and bound lifetime. Compared to this Y-type, they could only form this slip bound. And you can see that those mutants with a catch-bound curve, they still look pretty different, because the what we call peak bound lifetime at around 10 piconewton is very different. And we took this value called peak bound lifetime and found that the TCR signaling strength is actually correlated with this peak bound lifetime. So we kind of define this peak bound lifetime as the catch bound strength. So we think that the TCR signaling strength is really correlated with the catch bound strength. So how the different catch bound strengths affect these TCR downstream signalings. So we also look at this transcription factor activity in the T cells. So we collaborate with Ronjamin and the Webpan channel NIH. So they have pretty nice this transcription factor reporter in Jerker cells. So they look at this the ERK P38 and FAT activity in different TCR like TCR55 control or our two different catch-bound engineered mutants. So the conclusion is that they also found that different transcription factor activity is also correlated with this catch-bound strength. So it means that the catch-bound strength could also simultaneously regulate different transcription factor activity at the same degree. So I think the really interesting part or the application of this catch-bound engineering is how we could apply this catch-bound engineering on those tumor-related TCRs. So just like what I mentioned, that during the catch-bound engineering, we could do this high potency, low affinity evolution. So we could get lots of TCR with really high level effective signaling, but still similar low affinity. So we think that would be really useful for those engineering of tumor-reactive TCR and use them in TCR T-cell therapy, because the lower level affinity is always safer compared to this high affinity maturation. So we start this part by introducing a pretty famous story about the TCR T-cell therapy. So Dr. Carl Jung lead TCR T-cell therapy around 10 years ago. So they want to treat melanoma patients with TCR T-cell therapy. So they start from Y-type TCR with a really low affinity. So definitely not useful for the clinical trials. So they did this high affinity maturation to increase affinity for around 200 times and got this TCR called A3A TCR. So this A3A TCR could recognize this melanoma antigen called MAGE-A3. So this A3 TCR went to clinical trials to treat melanoma patients, but unfortunately, two patients died in the clinical trials because of this heart attack. And they found out the reason is that is a high affinity TCR not only recognize the tumor antigen MAGE-A3, but also cross-react with a peptide called titin expressed in heart tissue. So you can see the similarity between these two different peptides sequence. So it's really a reminder for people that high affinity maturation of the TCR is really it is potentially dangerous, because you never know whether this high affinity maturation not only give you higher signaling, but also cross-reactivity with self-peptides expressed in healthy tissue. So people really need a way to engineer TCR, not only enhance the signaling, but also try to keep the antigen specificity. So we think that the catch-point engineering probably be useful, because we could do this high potency, low affinity engineering for TCR. So we actually apply this catch-point engineering method to this case. So what we did is we look at this MAGE-A3 Peptide C complex with this Y-type TCR, and took the similar strategy to design a T-cell library, and did this T-cell display T-cell library and functional selection to really involve some TCR mutants with high level signaling, but still low affinity binding to these tumor antigens. So we got quite lots of mutants. So here is this experiment to show the activation with these tumor antigens. So you could see that the Y-type TCR is definitely really low potency TCR. You couldn't see any activation in the experiment. And this is the A3 TCR went to the clinical trials. It actually has highly enhanced activation compared to the Y-type. So we identify multiple TCR variants from the library. They also have highly induced effective signaling after the engineering. And we measure those binding affinity between MAGE-A3 Peptide C with these different TCRs. Again, we saw no correlation between these TCR signal strength and the binding affinity, since that we actually did this engineering not by high affinity maturation. So another challenge is that whether those TCR could cross-react with this known off-target toxicity from the titin peptide. So as you can see, this A3 TCR, which went to clinical trial, they could highly cross-react with these titin peptides. So that's the basis of this cross-reactivity toxicity in the clinicals. But for those catch-point engineered TCR from the library selection, we massively reduce this cross-reactivity with titin. And you could even actually find some mutants with absolutely no activation with this titin peptide. For example, this TCR, when we measure the affinity, you couldn't measure the affinity between this TCR and the titin cross-reactivity peptides. And the mechanism for this enhanced effective signaling against tumor antigen is by this catch-point enrichment. For example, in this catch-point measurement, we could see compared to the Y-type TCR, those engineered TCR we select as example 94A, 14, and 28AT, they could form this pretty high level catch bond with the tumor antigens with this peak bond lifetime almost like 4 seconds. And again, we correlate the TCR signal strength with this bond lifetime. And you can see the nice correlation between TCR signal strength and this catch bond strength. And we also test whether the tumor cells could be effectively killed by catch-bond engineered TCR T cells. So we use like tumor cells with this MAGE-A3 antigen positive signature for both melanoma cells and also colon cancer cells. So the conclusion is that our catch-bond engineered TCR could highly effective against the tumor cells in terms of cell death staining or cytokine secretion. And compared to this Y-type TCR. And one of the catch-point engine TCR, like 94A14, it could be even better compared to this high-affinity TCR, A3A, which went through the clinical trials. So last but not least is people will ask like, okay, this catch-point engineering definitely remove this cross-reactivity against titin, but how do you know this engineering method didn't introduce any new off-target toxicity? So we really establish a method to really comprehensively check the landscape of this off-target toxicity for any given TCR. So we use this method called is-display PMC library. So it's a library which could include 10 to the power 8 or 10 to the power 9 different kind of peptides. And you could actually throw in a TCR of interest and select against this TCR to enrich peptide which could bind to these TCRs. So in this way, you could know whether some of the cell peptide could be in the library to recognize to bind to this TCR. So in this case, we designed this actual AA1 restricted peptide library. So we basically randomize all other TCR facing residues into 20 different amino acids. And it's a library with functional diversity of 10 to the power 8 different peptides. So we did this peptide MHC library enrichment against either this high affinity TCR A3A or different catch-point engine TCRs. So the conclusion is that after this, the library selections, we could enrich some peptides and predict the self-peptide which could bind to those TCRs. So the results are actually pretty interesting, also kind of validate this method. So first, if you look at this high affinity mature TCR-A3A results, you could see in our prediction table, the titin peptide ranked #1 here. So it's actually a clear evidence show that it's really a useful method to identify any off-target toxicity peptide for a given TCR A3A, for a given TCR. So not surprisingly, in our enrichment, we could also enrich the known tumor antigen MAGE-A3 here. But for other catch-point engine TCR, we didn't see the titin in this table, but only the tumor antigen at a very MAGE-A3 at a very high ranking. So next step, we actually synthesize all these predict cell peptide and test them in activation screening experiment. So here is showing the showing the result for these high affinity mature A3 TCRs. The surprising thing is we found cross-reactivity with not only the known titin peptide, but we actually also identified two previously unknown off-target toxicity peptide called FAT2 expressed in small brain and also MAGE family protein called MAGE-A6. So it means that this A3 TCR actually have multiple off-target cross-reactivity and toxicity. So kind of validating our method. But for the catch-point engine TCR, the conclusion is that we only found the found this activation with tumor antigens. And for other predict cell peptide cross-reactivity, we didn't really see any real activations, suggesting that we didn't introduce any new cross-reactivity or off-target toxicity for those catch-point engineered TCR. So as a summary, we think that the formation of catch-point might be required for effective TCR PMC interactions. And the formation of catch-point could be engineered to induce effective TCR PMC interactions. And the TCR signaling strength may be correlated with these catch-point strengths. And the catch-point engineered TCRs could be highly potency, low affinity, and without off-target toxicity. And we think that this catch-point engineered TCR T-cell therapy will bring a new hope for the solid tumor treatment. So I will thanks to my my postdoc supervisor, Chris Garcia, for most of the work I mentioned here to support me to do this, and also thanks my new institute, Dr. Chen Jixu, Dr. Wei Yang, Dr. Guangchuan Wang, for their helpful suggestion and the support at my early stage of career. So also thanks all the funding agencies to support me at this early stage of work, and also my lab. So I'm happy to take any questions. Thank you very much.